Um, Hello, everyone. This is John Geller. I'm professor of invertebrate zoology and molecular ecology at Moss Landing Marine Labs. Welcome to our Thursday afternoon seminar. Before we start, I've been asked to read a short notice of rules of the road for the seminar. So firstly, all audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for the audience to ask questions live at the end of the talk, just like a normal seminar. Once the speaker has finished their talk, you can use Zoom's raise hand feature to notify the meeting host that you'd like to ask the speaker a question. The raise hand feature is located under the Participants tab at the top of the Zoom window. I'd like to mention also that at the conclusion of the question period at the end of the seminar, our speaker will hang out with us for a short while and we can have a more casual conversation that you're all welcome to stay and participate in. So it's my pleasure today to introduce our speaker, Dr. Liz Alter. Liz got her bachelor's degree at Yale University in organismal biology. She went from there to UC Berkeley and got a master's of science degree in environmental science, policy, and management. And from Berkeley, she moved to the crosstown rival Stanford and got her PhD in biological sciences, doing her work at our local neighbor, the Hopkins Marine Station. From Stanford, she did a postdoc at the Natural Resources Defense Council and then joined the faculty of York College at the City University of New York, where she was assistant and associate professor from 2010 to 2020. So she chose a very challenging year to move from the East Coast back to Monterey Bay and join the faculty at CSU Monterey Bay. Liz is a population geneticist. She works on phylogenetics and evolutionary genomics of fishes primarily, but she's also done research on historical genetics of whales, as well as the diversity of protists and bacteria in urban settings. And I also noticed she has a publication on the diversity of lichens, so quite a broad spectrum of interests and types of organisms worked upon. Today's seminar is titled Using Genomic Tools to Explore Fish Evolution in Extreme Environments. And with that, I'll turn over the screen, your screen, to Dr. Liz Alter.
Hi, everyone. Thank you so much to Dr. Geller and to Moss Landing Marine Labs for the invitation to talk to you today. Yeah, it has been a weird and challenging time to move across the country and switch institutions, but regardless of everything, I'm really looking forward to getting to know the Moss Landing community better and eager to collaborate and communicate with you all. So hope to meet you in person eventually, but in the meantime, I am always available for Zoom chats. So yeah, today I'm going to be talking about some of the work that I've been doing for the last decade or so on fishes, in particular in the Congo River. And just to kind of set us up here, as a biologist, one of the things that's always fascinated me is, the seemingly impossible places in which organisms can live. So there's, as you I'm sure know, there's a great diversity of fish species that can persist and even thrive in extreme habitats. So we have examples from notothynioid fishes in Antarctica, which use specialized proteins to persist in water that's below freezing. and then all the way up to fish in the, in the cichlid genus Alkalapia, which lives in hot spring derived waters in Kenya that are, 45, 40, almost 46 degrees. So really hot tub temperatures that we're talking about. We find fishes at great heights and great depths, from fishes in the genus Astyanax in high altitude lakes in the Andes. And then on the other end of the spectrum, all the way down to, snail fish caught at 8,000 meters in the Marianas Trench. So just an amazing extent of diversity of fishes that live in these extreme environments. And those environments, like the ones I just described, directly affect physiological functions, of course, which can lead to homeostatic imbalance or the inability to find food or reproduce. And so fishes that live in these extreme environments have evolved the ability to carry out their essential life functions by maintaining homeostatic balance, even in the presence of these extreme stressors. So they've gained or in some cases, as we'll see today, have lost traits that help them cope with and even exploit particular extreme niches. But so the question is, how do they do that? And more specifically, what are the genomic tools that fishes can utilize to do that? And are there commonalities across lineages? So, when we think about the evolutionary processes that drive the ability of fishes to diversify and adapt to extreme niches, we can think about the holistic picture. That is how processes of demography and speciation shape the patterns that we see at the genomic level. And in my lab, so as Dr. Geller mentioned, I work on a pretty wide diversity of things, but all involve genomics. I can say that. I used to be able to say everything I worked on was, lived in the water, but since the lichen work, that's not true anymore. But at any rate, we're interested in both these small scale patterns driving diversity in genomes, as well as, all the way up to large scale biodiversity patterns. And my lab does do work on environmental DNA and metagenomics as well. All the way up to biogeographic mechanisms that shape diversification. In the last 10 years or so, we've seen just this explosion in insights that we can get from different teleost species that have had their genomes sequenced. And these projects have shown some of the distinctive features of fish genomes that likely play a large role in fish evolution and in speciation. For example, the role of whole genome duplication. But it's important to point out that existing fish genome resources still lack a lot of important nodes in even in teleost diversity and evolution. For example, more than 80% of species that have sequenced genomes within fishes are in the Euteleosti lineage. So we don't have much info about many of the basally diverging lineages. And while we have great genomic resources in some clades like percomorphs, we have very scarce resources in others like osteoglossomorphs. So we're edging closer to good representation, but there's still quite a lot of work to do in terms of building out this picture of fish genomes that's going to allow us to better understand how fishes evolve in these extreme environments. I just want to mention a few of the projects that we're working on in extreme fish genomics, extreme fishomics. First of all, the project I'll be talking about today is this project on Lamprologus lethops, which is the only known blind cichlid. More about that in a moment. But we have a few more. We're working on a project on Danakillia, which is a genus of cichlid that lives in the Danakil Depression in Ethiopia and Eritrea. And so we're investigating the evolution of that clade using genomics. We work on the ubiquitous, at least ubiquitous on the East Coast, Mamachog, Fundulus heteroclitus, and how its microbiome may allow it to persist in places that other fish can't. And then most recently, we've started up a really fun project with a dear friend and wonderful collaborator at UNAM in Mexico, Jairo Arellave, on cenote-dwelling fishes and looking at cave and surface connections. So a real diversity of fish and environment. But to get back to the topic at hand, I'm going to be focusing today on this investigation we've been doing of this very, very, very odd fish. It's, again, the only known blind cichlid. So it's it's in this clade that's really known for its color and visual systems, but here we have one with no visible eyes and no pigment. And this project is a collaboration with these two wonderful folks, Matthew Artema, who's at Montclair State University, and Melanie Stiasny at the American Museum of Natural History. I'm going to do a very brief overview of the Congo system for those who aren't familiar. So it's shown here on the left. We're concerned with the lower Congo River, which is in this red box here. So this whole outline is the Congo River Basin, which is pretty enormous, a little smaller than the Indian subcontinent. So now we're zooming in onto the lower Congo. That is the stretch of river below Kinshasa and Brazzaville. So what I want to impart to you is that this is really one of the most dynamic stretches of river on Earth. As we travel along its length, we find these enormous rapids and cataracts and waterfalls and gorges where this massive volume of water that's being drained from the entire Congo Basin is forced through these really narrow chutes. So it's second only to the Amazon in discharge, but different from the Amazon because rather than sort of fanning out as the Amazon does, all this water is propelled through these narrow chutes, leading to some really just extreme hydrological conditions. We don't have a lot of information about the geological history of this river, this part of the river, unfortunately. But what evidence we do have suggests that the current high energy and high flow regime of the present day Lower Congo is recent and was probably not established till about 2 to 5 million years ago. Given how hydrologically complex it is, it's not surprising the Congo River Basin has a very rich fish fauna. And even though it makes up less than 2% of the overall basin area, as you can see here in this highlighted red patch, the Lower Congo has a very high proportion of regional diversity with about 340 recorded species, many more to be recorded. And then, about a third of those are endemic to this really narrow stretch of river. It's a very difficult place to work and has been pretty, poorly studied until now. So my collaborator, Melanie Stiasny, and I have been going for about the last 8 years. So this would have been our 9th summer, which sadly we had to miss. But we're now finally beginning the genomic analyses of a number of rheophilic or rapids adapted specimens that we have caught over the years. So this is just to give you a little bit of an idea of the hydrology of the river and how powerful it is in some spots. So, oops, let's see if I can play this video. There we go. So there's Ian for scale, you can see. So just massive, massive rapids. And the river is not all rapids. There are a lot of them. So these red stars indicate the presence of major cross-channel rapids. But we see that there's other complex habitats that are available to fishes, rocky shorelines, heterogeneous bed topology, a lot of turbulence, but a lot of sort of patches of calm as well. So we think these extreme hydrological features actually play a role in slowing both cross-channel and upstream downstream movement of fishes and other organisms, essentially creating population structure and diversification. This is another video that I'll just show just a few seconds of, again, just to give you, this was a extreme kayaker who did some sampling for the team. So you can see these are waves that you would not want to try unless you had quite some experience. Some of them are standing waves, like you could nearly surf on them. All right, so moving on to some of our findings, what kinds of fishes can make a living in these depths and rapids? So I'll talk a bit more about the cichlid in just a moment, but we've been studying this extreme hydrology of the river and how it affects divergence in several groups. And one of the most interesting has been this rapids adapted cichlid genus Teleogramma, in which we used next generation sequencing to detail a phylogeographic history. We used, so tens of thousands of SNPs to analyze genomes of fishes representing each of the different populations, and found that the ranges of these distinct lineages that you see here correspond well to geographic regions that are separated by rapids, by major hydrological and topographic barriers, indicating those features are probably major drivers of diversification. And what's unique about the Congo is that we see this diversification over these very small spatial scales. So sometimes distances as little as 1.5 kilometers. So pretty remarkable kind of micro allopatry. And we see phylogenetic lineages arrayed in a linear fashion along the river, again, separated by these major rapids in many groups, and a lot of them are cichlids. This one is the genus Lamprologus, which is a genus that is found both in Lake Tanganyika and along the Congo River, but this particular clade we've determined is in fact endemic to the Congo. And there's about 9 species that are endemic here, shown some of them, which raises some interesting biogeographic questions. Included among them is this unusual species that I mentioned earlier, Lamprologus lethops, the only known blind and depigmented cichlid. So it has only ever been found in this little tiny stretch of river, and we have a total of about 17 specimens at this point. So it's very rare, it's very difficult to find, and an extremely narrow endemic as far as all of our evidence suggests. So what we think is that, we have this fish that looks every bit like a cavefish, but the mystery is that it doesn't live in a cave to our knowledge. And moreover, it belongs to this family of fishes, again, that are renowned for their bright colors and the importance of vision in their mating systems. So sorting out where how this fish evolved and the genomic basis for its phenotype has been a real driving question for us. While most aspects of its ecology are pretty obscure, we do have some data on its habitat thanks to a USGS expedition in 2008 and 2009 that went to the lower Congo to ground truth and obtain depth and water velocity measurements using differential GPS and echo sounders and continuous depth recorders in this same little corner of the river where we find the lethops specimens. So, I mean, one of the things you can see is that the team recorded extraordinary depths. So some of the surveys measured depths of more than 220 meters, which is for a river. I know that doesn't impress you ocean folks, but for a river, that is extraordinary. So to our knowledge, it's the world's deepest, if not one of the deepest rivers. The data that you're looking here are ADCP data. So it's showing the visualization of primary and secondary velocities of the cross section of one location. And there you can see that there are these large persistent cells of upwelling and downwelling that send water shooting to the surface from these depths. So our hypothesis is that that lethops, this species, is living at these depths and occasionally gets entrained into upwelling events based on the fact that when we find specimens, they're typically dead at the surface or dying, and we have obvious signs of embolism and rupture. So it suggests to us that they are living at depth, and the reason we find so few of them is because there's not a ton that are getting brought up where we can sample them. The unusual phenotype of lethops is particularly remarkable when you compare it to the rest of the genus. So the rest of the genus looks like this. When we started this project, we had little in the way of phylogenetic information for the species, but our best guess based on morphology at the time for the sister was this species, Lamprologus tigropictolus. And you can see that this is, they are fairly similar in body shape, but that's about it. We now have some UCE data. So this is another project that's ongoing to build out the phylogeny of the genus using UCE data. So these are ultra conserved elements that help us to reconstruct the phylogeny of this recently diverged group. And in this analysis, it appears, so lethops is here in yellow, that it is not, in fact, sister to Tigropictolus, which is in pink here, but is rather sister to this entire lower Congo clade. However, I think this tree does give us some valuable things, including helps us to date the divergence between lethops and the rest of the clade. One other thing that I should mention that's quite interesting is that, along the way, we've also noted that the lower Congo has more blind depigmented species than just lethops. In fact, we see this same phenotype, in other words, cryptophthalmia, hidden eyes, and depigmentation and overall body depression, along with some other features. We see that this same phenotype across distantly related evolutionary lineages. So here we have a pretty nice case study of convergence. Convergence is, of course, typically thought to be adaptation to shared environment or selective pressures. And the mystery for us remains what that selective pressure is. Because not all of these species are found at depth the way that we believe lethops is. So some of them we catch in great quantities at the surface. So that's another mystery. You'll note here that we have two different species of spiny eel that are both depigmented, and shown on the left are the closest pigmented relatives that we have. Of those spiny eels that we find in the lower Congo, actually we find 4 depigmented species, and then 2 other endemics that are pigmented, and then a bunch more that are found in the lower Congo but that are not endemic. So one of the early questions that we asked was whether we see convergence within this genus of spiny eels, since we have multiple cases of this phenotype. And we did a quick and dirty phylogeny to figure that out, and found that, in fact, contrary to our expectations, the phylogenetic evidence demonstrates that cryptophthalmia, this cavefish-like phenotype, evolved at least twice within this genus. So this is a pretty good indication to us that whatever this selective pressure is in this river, it is active and it is strong. It suggests a strong role of selection in shaping this phenotypic diversity, and part of our project involves anatomical analysis using CT scans to actually quantify morphological differences in these taxa. And when we do that, we find that we see obvious differences in the size of the orbitals and some of the those more obvious features, but we also see some other features that we might not have predicted. For example, hypertrophied jaw musculature. We also see enhanced lateral line, as you might expect in a species with no vision. So this is just another kind of close-up on lethops to zoom in on our cichlid here. We've started some in-depth morphological characterization on some of the so-called degenerative features of the eyes and other aspects of the anatomy, including these very shortened optic globes, as you can see here in the E panel, a decreased number of neuronal layers in the retina. We find absence of extraocular muscles. One other bizarre thing, and maybe this is something for those that work on marine fishes that could help us think about, we see a very thickened wall of the swim bladder here compared to Tigropictolus, which essentially you can't, in this scan, you can't even see the swim bladder wall. So just sort of a interesting side note that we're curious about and particularly thinking about in terms of its relationship to depth. Okay, so to get to the genomics then, we've started with, so when we started, we had pretty good cichlid genomic references for the genus as a whole, and in particular, a recent genome of a sister clade, Lamprologus pershardi. And we sequenced lethops as well as, again, what we thought was the sister at the time, Tigropictolus, and with the goal to understand how this species genome, how lethops' genome differs from its congeners that have these non-cryptophthalmic phenotypes. So that have what we call, normal fish phenotypes. And we also wanted to investigate whether we see a convergent set of genes evolving in the other sequenced cavefish, Astyanax mexicanus, so the true Mexican cavefish. And finally, we were interested in understanding whether trait loss in this lineage evolved neutrally or was selected for. So to do this, we sequenced genomes of lethops and tigropictolus and mapped them to this annotated referenced cichlid genome. And our comparative strategy here, which is what we're hoping to use for these other species and kind of build on, has this three-pronged approach and exploits the excellent genomic and transcriptomic resources for these two additional species, the Mexican cavefish, Astyanax, and then zebrafish. And the first and simplest thing we can do is look across genomes for candidate genes. So numerous genes that have been identified in other fish species with unusual phenotypes, such as MC1R for pigmentation, for example. And we can look for genes with mutations of large effect, those mutations that obviously disable the gene, so stop codons, frame shifts, et cetera, and then take a closer look at those genes to see what function they perform. And then finally, we're able to compare across the genome overall, synonymous and non-synonymous changes in lethops compared with tigropictolus. And so throughout, we're using this comparative approach wherever possible, in particular, again, utilizing these relevant fish models for which we have good genomic and transcriptomic resources. So to start with, just some results, what happens when we look across genes that show variants of large effect, for example, indels or stop codons. So if we look at the distribution of functional categories that these genes fall into, we see something interesting. So here we're looking at the top 10 categories of gene ontology terms that are enriched in lethops on the left and tigropictolus on the right. And you can see that these categories are quite different. And in particular, we have a few nice categories that pop out that are related to eyes, as well as a number that deal with neural and sensory system development. And we really, the question that we always get is about transcriptome for lethops, and we're working on it. We're trying. There is a hobbyist in Montreal who's been really working to get live specimens of lethops. Again, we always find them either dead or dying, so we've not yet been successful in getting RNA. But if this hobbyist is able to successfully breed them, then that will be very exciting for everyone. So, when we compare these, this list of the top 10 categories of Go terms to the one in L. tigropictolus in our sisters, our related species, we see instead a list that includes a lot of genes related to environmental responses or immune function. So no eye genes in there. So we decided to take a closer look at some of these genes, and I'm just going to give you a real quick tour of just a few of the interesting ones. We first filtered those genes to find those with variants of large effect, and then we screened those for a Go term that's relevant to the phenotype that we are especially interested in, including eye-related genes, metabolism-related genes, circadian rhythm, pigment, all aspects of phenotype that have been shown to differ in cave fishes. And then we combed the literature for genes that are shown to have a strong phenotypic effect when mutated. So a pretty stringent filter, and definitely not perfect, but a place to start and to kind of anchor our study. And so when we do this, we end up with this shortened, but by no means short list of genes to examine, with eye genes being the most abundant, followed by metabolism. And I just want to give you a few examples of some of what what we've uncovered as we've gone through this list of genes. So I mentioned MC1R before as being important in pigmentation, and we did look at that one first, but we found no changes between MC1R in lethops versus tigropictolus or Lamprologus pershardi. So same sequence essentially. However, the one that did emerge is this gene, the OCA2 gene, which provides, that particular gene provides instructions for making a protein located in melanocytes and is pretty critical to the pigmentation pathway. So a lot of solid work in Astyanax and cavefish shows mutations in this gene are responsible for pigment loss in actually a number of populations that have evolved it independently in the case of Astyanax. And what we see in lethops, as you can see here, is actually a loss of the start codon. So that should be completely sufficient to prevent the OCA2 gene from being expressed. We also see a number of crystalline genes that have mutations of large effect that are disabled in some way. So these are structural proteins that lend lenses in the eye their transparency and integrity. And you can see, for example, here we have gamma crystallin and B gene, and we see a frame shift mutation. So we have a number of these crystalline genes that show mutations. None of them are the same mutations that we see in Astyanax, but Astyanax likewise shows a number of changes in crystalline genes in general in that gene family. So, kind of, those are some examples of some of the genes that we've found. And, I just wanted to kind of zoom out to a broader evolutionary question here. So we've shown that we do, in fact, find that genes with variants of large effect are enriched in Go terms relevant to the phenotype in accordance with our hypothesis. And also that some of these genes may be, convergent with Astyanax, the cavefish, and maybe others aren't. But what about this, the question about whether regressive traits are neutrally evolving or adaptive? It seems like a simple question at first, but as you delve into it, it's actually quite interesting and complex. The regressive phenotype of cave animals has been a debate among evolutionary biologists since Darwin, who said, as it's difficult to imagine that eyes, although useless, could in any way be injurious to animals living in darkness, I attribute their loss solely to disuse. So he was solidly on the neutral side of things. But after neo-Darwinism and the rediscovery of genetics, evolutionary biologists have thought more about this issue, and many have attributed the loss of eyes and pigment in cavefish to actually adaptive evolution because of the fact that eyes and pigment are expensive to produce. But we haven't had fantastic support for either mechanism. But now with new genomic approaches, we're well positioned to make some progress on this question. And we can ask things like, what kinds of signatures would we expect to see in the genome under these different scenarios of neutral evolution versus adaptive, and whether our observations would support either hypothesis. So, that is one of the avenues that we are exploring right now. And one of the first ways that we wanted to explore it was by getting an idea of the basic demographic history of these species. So if either of them underwent a tremendous bottleneck, then that alone could be responsible for the fact that we see certain of these alleles that have persisted in the population, rather than it being a necessarily adaptive. So, here we're using a new analytical method that lets us model changes in effective population size through time using information from whole genome sequences. So this is the PSMC pipeline. And it also infers the time to the most recent common ancestor for each independent DNA segment, and based on the rate of coalescent events, infers ancestral population size through time. So what's cool and what we see is that we have a very different pattern for our two species. So we're looking back in time. So this is, a million years ago or so on the right-hand side of each graph. What we can see is that lethops underwent this tremendous bottleneck, very, very extreme bottleneck, and has maintained a very low population size ever since. So, we suspect that this is the initial kind of speciation event, and that the population has remained at a low effective size since that time. Tigropictolus also goes through a bottleneck, and then its population size recovers subsequently. So, again, we feel this suggests that, at least in the case of lethops, some of the reduced genetic variation, some of those alleles that we see, they may have been fixed due to this bottleneck. That's something that we can't discount at this moment. So we decided to try another tactic that should be a bit more sensitive to recent evolutionary changes and look at heterozygosity across the genome. We're a bit limited in that, in the moment we have genome data for only one individual of lethops and only several of Tigropictolus, although we just got data back for an additional 8 specimens of each. And here we're using a sliding window approach to look at how heterozygosity changes near our genes of interest across different scaffolds. So we're leveraging this species comparison to see if the species look the same or different at around regions of our genes of interest. So we first of all observed that, as expected, overall heterozygosity in lethops is much lower than Tigropictolus. So here on the left here we have our heterozygosity, and it's essentially 0 around these our genes of interest. But we can also see these dips that occur in lethops in heterozygosity that do not occur in Tigropictolus. So, that's pretty cool. Here we're looking at just 3 of our genes of interest because they happen to all occur on the same scaffold. This is DDB2, a UV repair gene. I'll mention again in just a second. This is a metabolism-related gene, and this last one is a crystallin related to both eye loss and circadian rhythm. So for all of them, we see this trough of heterozygosity near the position of the gene itself, and also like the surrounding region, which suggests a selective sweep. So this is something that we're following up with our additional genomes to see if we have this pattern when we look across additional genomes. We do see the same thing when we look at other genes of interest on our other scaffolds. And you can see up here, we don't see that same pattern of troughs of heterozygosity around our genes of interest in Tigropictolus. So, that's to us some evidence too that these eye loss and other alleles were not just evolving neutrally, but in fact were selected for. So we now have a decent population sample built up, like I said, that we've sequenced. So we'll be looking at population-wide heterozygosity around those loci. So these analyses together suggest that it's not an either-or question, either neutral or adaptive, but that likely both processes are playing important roles in shaping the evolution of this very strange species. All right, I just want to close out by highlighting an interesting aspect of working with genomes from rare species that we have very little ecological information about. In a case like Lamprologus lethops, we find ourselves making a lot of guesses about its habitat and its ecology and its life history because we can't sample it directly. And this is true for a lot of marine species and freshwater species as well that may be rare or just very, very difficult to sample. So, one of the points that I want to make is that having a genome can actually be an extraordinarily rich source of information about these species that allow us to make some inferences or at least build hypotheses about basic biological aspects of the species that we haven't been able to get any other way. So, for example, we hypothesize based on the appearance of lethops that it lives at depth in a lightless environment. But we don't have any proof of that, as it lives in a river and it could be migrating up and down. So when we began, we weren't looking specifically at UV damage repair genes as an interesting category related to phenotype. But then a number of those genes emerged in our analysis as having these variants of large effect. And when we looked closer, we found there were a number of these genes, such as this one you see here, DDB2, that seem to be pretty essential for repairing the incessant damage that comes with UV light. So that's not to say lethops might not have another way to fix DNA damage from UV, but we consider that, we're considering this to be further supporting evidence that this species is not, this population is not exposed to much, if any, UV light on a consistent basis. Likewise, we were curious about whether they share a distinctive trait with Mexican cavefish, despite the fact that their environment is pretty different. So cavefish live in dark and pretty nutrient poor environments, and they tend to be remarkably starvation resistant. So here you see the blind morph compared with the surface morph. Once a year or so, when food gets swept into the caves by these floods, they binge eat. And so they've been a really interesting model for metabolic genes because of that. We don't know if the lethops environment is similar in that regard. We don't know really much about what they're eating because they tend to have emptied their stomach contents by the time we find them. And we don't know if their environment is very nutrient poor, though we guess that it is based on the nutrient status of the river as a whole. But here's what we found. We found numerous mutations related to the loss of appetite suppression. So one interesting example here is spexin. It's a neuropeptide for which we know quite a bit about its effects on food intake in both fish and mammals. And this is a gene that's been disabled in lethops. So it's got a loss of function mutation. And that suggests that in fact, their environment may be nutrient poor. Subsequently, we looked at some of the CT scans from lethops and Tigropictolus, and we find the lethops do have substantially greater fat reserves than Tigropictolus does. So that's another avenue that we're going to be examining in more detail. And curiously, that this spexin is a different mechanism than has been found in cavefish. So Nick Roner and some of his colleagues at STARS have analyzed genome sequences of these cavefish, and they found mutations in a different gene. This is the melanocortin 4 receptor, which has been linked to anorexia in humans. That receptor is regulated by insulin and an appetite suppressing hormone as well. So here we have the same phenotype, but different genes that appear to be affecting it. All right, so what's next for this project? We're continuing to analyze the genomes from Lamprologus, from lethops and its congeners, and we still have a lot of work to do on this genome. So we published one paper that was sort of the low-hanging fruit, but we'd like to look at regulatory regions, gene duplications, genomic architecture, and then we've started sequencing additional specimens from these other lineages. So we started with cichlids because of the great genomic resources, but we've got a myrmidid that we've sequenced now, one of the mastacembelids, and on our 2017 expedition, we actually pulled out this, we only have one specimen of it, but it's a pretty cool fish that looks to be sister to lethops, and it's sort of got an intermediate phenotype, which is really neat. Okay, so I'd just like to end by noting that these projects are really about understanding the fundamental ways in which genomic and phenotypic diversity on our planet evolves. But the extreme habitats that have generated all of these species that I mentioned are nearly all under threat, often even more so than your average habitat, whether due to climate change or pollution or other habitat destruction. And freshwater ecosystems are very vulnerable because of their proximity to human populations and overuse. So, for example, in Congo, a massive dam is in the works, planned for construction, called Inga 3, which would have more than double the capacity of the Three Gorges Dam in China, which is currently the largest energy generating body ever built. So we still don't know what's going to happen with this, with COVID and World Bank funding, et cetera. But they're all indications suggest it is going forward, and it will have massive and catastrophic impacts on both the local human population in this area, as well as ecosystems that are home to this extraordinary array of fishes. So, as we lose these important habitats, we also lose the evolutionary libraries contained within them. with each species representing millions of years worth of information about how organisms solve these thorny problems about persisting and thriving in their environments. So just a little conservation message there at the end. And with that, I'd like to thank my collaborators and our funding sources. We've got an amazing team of collaborators in Congo without whom this work could never happen. And thank you for your attention, and I would be more than happy to take questions. So I'm going to stop sharing, I think, yeah. And
okay.
Start video. There we go. Oh, I can't start my video because the host is okay.
Do you hear me,
yes.
Liz?
Yes, I do.
Okay.
There we go.
Okay, so yeah, I'm going to moderate the question answer period. And so if the audience could use the hand up symbol on your in the list of participants you can access on the right side of your panel, I can call on people. So while we're waiting for people to think of their questions, I have one. So you showed us your phylogenetic tree for Lamprologus, and you showed us that lethops falls in a very basal position. That seems kind of odd to me, or just maybe it's just one of those things. But it's like the most divergent, at least in these phenotypes, and yet it falls basally. So since we probably would assume that blindness is not the ancestral condition, it attained that sometime after it split off from all the rest of quite a number of species there. And then I guess couple that to your demographic history analysis, which shows, I forget the time estimate, but I mean, not hugely long ago, so that's a lot of evolution in the rest of the genus that happened after lethops branches off. And then in that same period, lethops doesn't spawn any other sisters, and also goes through this really dramatic phenotypic change. So it's sort of it's kind of a peculiar phylogenetic setting, which I guess I don't have a real specific question about it, but it struck me.
Yeah, no, and you were paying very good attention to pick up on that, because it was also a big surprise to us when we saw it fall out basally. And we're not absolutely convinced by the UCE tree yet. I mean, as I'm sure you know, there's like 1,001 ways to run those data, and depending on whether we use Astral or various species tree building platforms, we get slightly different results, and sometimes lethops does not fall out basal. I think what's going on is that the radiation was very rapid in the lower Congo, and that's causing us real difficulties in reconstructing that clade. The reason we went to UCEs was because we tried all the usual phylogenetic markers first, and got essentially gobbledygook, no resolution. We're really hoping, so now that we have the genome for that one specimen that I showed, which is, we're thinking of it kind of like, missing link is not quite the right word, but we do think it's sister to lethops, and we're hoping that is really going to help us to resolve the tree in a way that we haven't been able to quite so far. But it does look to be very, very like the clade diversified very quickly into a number of different niches. And the other mystery is when you layer on the biogeography, we find lethops in this one little spot, whereas the other species within the genus are arrayed in a relatively nice kind of linear fashion down the river, that suggests kind of serial colonization, almost like you would expect. So some deviations, but that suggests the scenario that you mentioned, which is, we had that the ancestor of lethops was more widespread, and then it went through this bottleneck as it specialized into this very specific niche in one spot in the river. So yeah, more to come as we as we get the genome of the sister species integrated into our UCE tree.
Yeah, I guess I don't know how you feel it might have affected your in-depth genomic analysis, because as it turns out, your point of comparison is not as close as you anticipated. And when we look at the other tree you showed us for, and I forget the genus and species, but other blind fishes, they were all they all had a sister, very so it's something that can evolve rapidly and multiply. So there, at least in that other genus, there's an opportunity for a comparison to a very good candidate of the sister, and really not have to work your way back through so many evolutionary nodes to get to the point of comparison.
Absolutely. No, you've hit the nail on the head. I mean, the reason we started with the cichlid as our first dive into comparative genomics was because we have so much in the way of resources for cichlids as a group that we don't have, for example, for like more myrids or certainly mastacembelids. But the phylogenies for those are a bit better worked out, and they don't seem to hybridize like crazy the way cichlids do, which is part of why there's such a headache when it comes to reconstructing their species histories. So we see hybridization all over the place in all the cichlid groups that we examine.
Okay, we have a question from Scott Hamilton. I don't know if, okay, he's unmuted. So.
I was just unmuted, so yeah. Thanks, Liz. That was a really fascinating talk, and the fieldwork just looks so exciting. You put out the call to have people think about why lethops has that thicker swim bladder, right?
Yeah.
So I started thinking about it and chatting with one of the other students who was watching, and so I just wanted to run a couple ideas by you and see what you thought. I mean, I was thinking two things. I mean, potentially, right? You said that the river seems very turbulent, right? And sometimes they get sucked up in those upwelling cells. So I was wondering if maybe having that thicker swim bladder wall might help prevent rupture, or swim bladder rupture, if they're constantly getting sort of sucked up and then have to sort of force and find their way out and back down again.
Right.
And then I was also thinking about maybe there was an auditory function, since the swim bladder can be used to amplify and enhance sounds, and they don't have their visual resources. Maybe that thicker swim bladder wall, sort of maybe holding the swim bladder more rigid, could, enhance vibrations or something. So yeah, who knows? But that was kind of where my mind went, thinking about those things.
Yeah, that's a really interesting thought. We had been wondering about the entrainment and upwelling as well, and what we don't have a good sense of is how often those events happen, and whether it's enough that that thickened tunica on the swim bladder would really help. Well,
you don't want it to happen more than once before you mate, I guess. So
yeah, exactly.
It wouldn't have to be all too frequent for there to be a selective advantage.
That's very, very true. Yeah, I mean, a lot of the other direction that we see cichlids in and fishes in the rapids go is to lose the swim bladder altogether.
Right.
So that's also kind of an interesting.
Yeah, why does it still have it, right? If it is
right
really down near the bottom and doesn't want that to happen, yeah.
Yeah. Exactly.
So maybe there's trade-offs then with these other functions of the swim bladder.
Yeah, and the sound one is particularly interesting. I mean, we've also been kind of curious about the lateral line, the enhanced lateral line, and the fact that you do expect that. You see that in cave fishes, not just Astyanax, but a lot of cave fishes. And so at first we're like, oh yeah, that makes sense, until we remembered, these guys are living in extremely turbulent conditions, and wouldn't having an enhanced lateral line just be like, it would be like,
or it would be firing.
It would be like you have your hearing aid turned all the way up all the time or something. Yeah, so that's another kind of just big question mark. We have a lot of them, clearly.
Really neat. Thanks.
Thank you.
This is John. I had another question, or I don't know, an observation. So one of the genes that you talked about a lot was Crystallin. And I remember that when I was a postdoc in Denny Powers' lab at Hopkins Marine Station, that to my surprise, I learned that Crystallin is actually I don't know what the term is, an isoform of lactate dehydrogenase, which was the workhorse, the enzymatic workhorse for the Powers lab back then.
Right.
In Fundulus.
Yeah,
in Fundulus,
it all comes back to Fundulus.
Yeah, and so this was really illuminating for me back then, because I was coming from a pure ecology background, that the protein lactate dehydrogenase, which is a catalytic enzyme, and crystallin, which is a structural protein, which is used in eye lenses because in crystalline form it's transparent, are the same. They're the same protein, which is cool. It's cool, and it kind of opens your mind to how how evolution can occur in kind of in co-opting ways in the genome. But it also causes me to reflect on your study, that and I don't know if it's the same crystallin that we're talking about, but a gene can produce products that have completely different functions in an organism, and so it would be bad to lose LDH even if you didn't need crystallin, right?
Absolutely, yeah.
So that's sort of a constraint on both selective change, if a gene sort of functionally protected by its involvement in some other pathway, and also against drift, right?
Yeah, that's right. Yeah, so I mean, we've sort of considered those pleiotropic aspects quite a lot as we looked at candidate genes, and we asked, well, why hasn't this thing accumulated mutations, right? Like, we know that this particular gene is involved in maintaining the retina in some way. Like, why has this one but not this one? And so that's always not being a proper cell biologist. We're always trying to delve into the literature and probably go places where we shouldn't. But with the crystallins, I mean, one of the things that I was amazed to learn when I started trying to educate myself on eye genes and eye development genes is just the extraordinary diversity of the crystallin gene family. It's a real big gene family. So there's a lot of room for experimentation and for things getting lost and messed up there, which is not true for something, like MC1R, we find to be intact completely, not even a handful of non-synonymous substitutions. So, that's a case in which there's surely some other, we know there's lots of other functions that MC1R, lots of other roles it has. So, but yeah, the pleiotropy is always the number one thing that we have to think about when we're considering loss of function. That's a good point. I'm glad we managed to turn the conversation back to Mama Chogs.
Well, I'll give it just a couple more minutes in case someone has a question that's formulating. Can I ask just a technical question? Like when you see like a single nucleotide insertion, like you did in one of the alignments you showed us, how are you confident that that's just not sequencing error?
You sound like one of our reviewers.
No, that was me. I'm just kidding.
Yeah, no, we did two things. First, we went back and sang or sequenced them all, like all the big ones, all the ones that we were really highlighting in our paper as important. We went back and sang or sequenced with in other specimens of lethops and tigropictolus. And then we also, we stacked up our reads to show that we had, what coverage we had for each of those regions, to make sure that we had, that we weren't basing it off of really low coverage at a certain spot.
Okay.
So yeah.
Well,
yeah, that turned out.
That satisfies me. I guess it satisfied your reviewer too.
Yeah, the lesson is don't throw out your old ABI. Still need it.
Okay, well, I don't see any more questions. So I'd like to thank you for a really great seminar. That was really interesting. I really enjoyed